Hi, Dr. Windisch from Sparks Pediatric and Adolescent Medicine, coming to you from somewhere in the deep south. Today what we want to talk about is viruses, virus particles, and what makes them different from other germs that we've run across, and then specifically how this plays into the coronavirus infection that we're all sort of dealing with. My goal here is to teach you just a little bit of basic virology, because I think if you understand viruses and coronavirus in particular, it'll kind of demystify some of this, make it a little less scary, and you'll understand what we're talking about. So we're going to turn over to my white paper here, and Benny, my faithful assistant, is going to zoom in, and I apologize for the low-tech way we're doing this, but we're having some tech issues. So first, we want to take a look at, get an idea of scale. So if we look at a red blood cell, a red blood cell is 6 to 8 microns. I think I got it. There we go. Okay, which means it is 0.006 millimeters. Yeah, okay. So if that's roughly a millimeter, you get an idea of how big a red blood cell is. So if this is a red blood cell, and this is roughly 6 microns, keep that in mind. All right, group A strep. The bacterium responsible for strep throat is 2 microns. Okay, when you look at a bacterium under a microscope, you have to use what's called an oil immersion lens. The microscopes you had in high school did not have oil immersion lenses unless you were at a really expensive, really fancy high school. So you need special lenses, and even then, quite honestly, they're hard to see. It's real easy to miss them unless you have a trained eye. The limit of resolution on a microscope, the smallest thing you can see, and this is on a light microscope with white light, is about 0.2 microns. So if this is 2 microns, you can see that little black square. If I blow this little black square up onto the next page, we'll start with making that little black square about the same size that it is here. Coronavirus is 0.12 microns, or roughly that big. So if we compare this to an RBC, you can see how small it is. And realize if this is an RBC, then a human being is bigger than my 2-story house. So, you know, we're really coming down here in scale. So 0.2 microns or 120 nanometers. Okay, so in order to see this, you need an electron microscope. Can't even see it with a light microscope. To put this in a little perspective, polio virus, which we have a vaccine for and was one of the first things we developed a vaccine for, is 30 nanometers. So roughly a quarter of the size of that. So would be roughly that big. So if I drew coronavirus this big, poliovirus would be that big. Okay. Now, what is the structure of coronavirus? Proteins or viruses are not living creatures. They don't breathe, they don't eat, they don't take in nutrients, they don't produce waste products. What they do is they are sort of bots that get inside of a cell and co-opt the machinery of the cell to make more of themselves, but they use the cell's machinery to do it. So coronavirus, this little corner here, will look something like this. It has a layer of fat. We call that the lipid bilayer. And it's essentially made up of the same stuff that vegetable oil is made up of. Okay? And then sticking out of this lipid are several proteins. The first one is what's called the spike glycoprotein or the S protein. There is something called the membrane protein or the M protein. There is the hemagglutinin protein. Looks kind of like that. There is the E protein. And they kind of stud the whole cell. When you see the pictures of coronavirus, the big studs that you see are the S protein because they're pretty big. But these other proteins exist coming through this layer of fat as well. Inside of the virus, and this isn't really a cell, it's just a virus particle, we have the genetic material. Viruses can have DNA as a single strand or double strand. They can have RNA as single strands, double strands. They can have multiple strands of either DNA or RNA, but they don't have both. Okay, this particular virus, its nucleic acids come in the form of RNA. We're going to talk about what RNA is in a minute. And the RNA is studded with another protein called the N protein, or the nucleocapsid protein. And it sort of forms a protein layer over the RNA. All right, and these studs are going to be really close together so that you can't really see in between them. RNA and DNA both come as both sense or antisense, and sense is written as a plus, antisense is a minus. This is what's known as sense RNA. So what does that mean? Well, let's start with DNA. We have 23 pairs of DNA, or we have 23 chromosomes in pairs that make up our DNA. And if you look at them under an electron microscope, they're essentially two strands. One of these strands encodes the gene. The other strand is a backup copy that's reverse of the strand that we use to make stuff from. The strand that we make stuff from is sense or positive. The backup strand, which is backward, or the negative of the reading strand, is called the antisense. This is here in case we develop an error when we're making copies of the cells. It's a backup and we can do repairs from it. When we go to actually make something from this, we peel the proteins, or we peel the nucleic acids apart. RNA polymerase then hops on to the plus sense, or the plus or sense strand of DNA, and it reads it, and it makes sense RNA, which is a single strand, and it'll make multiple copies of the RNA from this gene. These then exit in human cells, exit the nucleus, go out into the cytoplasm of the cell, where they are read by transfer RNA and ribosomes, which contain RNA as well, and then they're translated into proteins. Okay? Viruses that have antisense RNA, before they can do anything, that RNA has to be converted back into sense RNA. So there's an extra step. But once this gets into the cell, because it's sense RNA, it's plus, the human cell, the ribosome hops on and just starts reading this. And as it reads it, it starts churning out proteins needed to make the virus. Those proteins, at the same time, other proteins will copy this for the virus. And then the parts just self-assemble. When they self-assemble, the proteins and the RNA, they take a part of the cell membrane from the human cell with it. That's where it gets its lipid layer. It steals it from your cell and goes out into outer space to go on to infect other cells. So that's kind of how this protein works. To put it in some perspective, poliovirus has one strand of DNA, and I don't remember if it's DNA or RNA. I think it's one strand of RNA, but it's much smaller, only makes a couple of proteins, and is much easier to just make a vaccine to. What the vaccines do is they'll take this part of the protein, or the hope is you'll take this part of the protein, you'll introduce it to people, and they'll make antibodies to attach to that protein and block it from doing its job. At least that's the hope. We're not there yet. We know what the proteins are, we just don't know that we can actually make effective antibodies in a vaccine and do it in a way that's safe. Anyway, so that is kind of the nature of the beast that we're up against. I hope that, I know there's a lot of molecular biology and a lot of big words, but my hope is that this will kind of put some perspective for you so you understand what we're up against and kind of demystify stuff. So that, you just kind of know where we are. So once again, this is Dr. Kevin Windisch from Sparks Pediatric and Adolescent Medicine, coming to you from the middle of the coronavirus pandemic somewhere in the deep south. Stay safe out there, everybody. We'll see you next time. If you have requests for more information on this or other medical topics, please put it in the comments, because I do take requests. So please feel free to request. Please like this video if you liked it. Please click on our sponsors. They allow me to keep doing more videos. Thank you.